I research how a crop disease is spread by insects. Let me introduce you to the blue-green sharpshooter. It's one of the major insects that spread Pierce's disease of grapevines, which cost California over $100 million per year. The disease is caused by bacteria that survive in the foreguts of the insects, and as the sharpshooters feed on plants, they spread the disease. Blue-green sharpshooters feed on grapevines by piercing plant tissue with their proboscis, which is indicated by the arrows and shown on the next slide. This diagram illustrates the proboscis piercing plant tissue during insect feeding. The insect sucks plant juices into the foregut and then spits them back out. It's during this process of repeatedly sucking in and spitting out that the disease is thought to be transmitted. Scientists and engineers are researching this process with the hope of finding targets to prevent disease transmission. I predict that fluid dynamics during the spitting out process play a major role in Pierce's disease transmission. I created a 3D model of the insect foregut and simulated the fluid dynamics of spitting. My results predict that there are regions in the foregut with drag forces high enough to detach bacteria. Once the bacteria are detached, they can be spit into the plant and cause disease. And that's not the only reason I have for a flow-driven transmission hypothesis. There's also the bacteria colonization patterns within the foregut, which mirror fluid dynamic predictions. But that's actually hard to illustrate at the current magnification of the model. There's a region of the foregut that is specifically linked with bacteria transmission. It's composed of the magenta and teal portions of the model. Now, the bacteria attach to the inner walls of the foregut, which are hidden in the current view of the model, but they can be visualized by taking molds of these magenta and teal sections. These two molds here, they're 3D molds that represent the segment of the model that's of interest. It's been studied more closely in previous studies where they examined the bacteria transmission. And I plotted my simulation results within those molds and obtained these simulations here. These are rainbow colored heat maps where the dark blue regions indicate locations with low predicted drag forces, yellow indicates regions with high predicted drag forces, and dark red indicates regions with very high predicted drag forces. Now, let's compare this with locations in the foregut where bacteria were observed soon after they attached. That's what's represented in these rectangles, which I derived from the reference down at the bottom of the slide. And the blue circles in the rectangles indicate locations where bacteria were often observed. Yellow indicate locations where bacteria were seldom observed, and the blank regions indicate locations where the bacteria were never observed. And I should say that this rectangle on the left corresponds with the simulation on the left, and the rectangle on the right corresponds with the simulation on the right. What I want you to notice here is that bacteria colonization patterns mirror fluid dynamic predictions. For example, in the rectangle on the left, down at the bottom, there's this cluster of blue circles that corresponds with the region in the simulation on the left with lots of blue in it. Similarly, in the rectangle on the right at the bottom, we have a cluster of darker colored blue circles that correspond with the simulation on the right, the region in that simulation with the lowest predicted drag forces. Now, just above that region is a section that kind of looks like a saddle. And in the middle of that saddle, there are higher predicted drag forces, and then above and below the saddle point, there are lower predicted drag forces. And these can be viewed in comparison with the rectangle on the right, where there's this saddle-like colonization with low colonization frequency in the middle, and then higher colonization frequencies above and below that potential saddle point. Furthermore, at the top of these rectangles, we see that there's lower predicted colonization there's low observed colonization here with some yellow and lots of empty space, and this corresponds with regions in the simulations with high predicted drag forces. So to summarize this slide, what I'm saying is that the bacteria colonization patterns mirror fluid dynamic predictions, and that there are yellow and red fluid dynamic predictions that are higher than the drag forces required to detach bacteria. Therefore, fluid dynamics likely play a major role in bacteria detachment from these segments of the foregut. And I want to contextualize that with a slide that we saw earlier. What I'm saying is that in this region of the foregut, it's likely that fluid dynamics play a major role in detaching bacteria, which then get spit into the plant to cause disease. And if you remember when I talked about this slide earlier, I talked about how the insect repeatedly sucks in plant juices and then spits them back out. Now, this is part of the insect's ritualistic feeding behavior that's thought to be about the insect tasting the plant juices. So I hypothesize that if the insects were to encounter plant juices that were more tasty to them or more suitable to them, then they might spend less time in that process of sucking in and spitting out. Less bacteria would be detached from this region of the foregut, and less bacteria would be transmitted. So it may be worthwhile to seek out plant juices that are more suitable to the insect. And that could be counterintuitive to a lot of the viewers, because why would a farmer want to create a more welcoming environment for these insect pests by fostering plant juices that are more suitable? Because feeding preference is expected to be less important than pesticide application for reducing vector populations in commercial settings. So the idea would be to keep the insect comfortable and happy until it dies from the pesticides, so that in the meantime, it's not transmitting as much bacteria. Okay, so the next question would be, how do we make these plant juices more tasty to the insect? And there's at least two variables that have been shown to be correlated with the insect feeding rate. Those are the chemistry of the plant juices and the fluid pressure of the plant juices. And these variables can be influenced by watering treatments or fertilization treatments, which could be simple and cost-effective methods of combating Pierce's disease transmission. If combating the disease could be as simple as adjusting irrigation settings or applying fertilizer, then farmers could protect their crops using practices that are familiar to them. Perhaps it's time that trials be conducted to examine the effects of watering treatments and fertilization practices on Pierce's disease transmission. Thank you.